from BDS first year studying in Frontier Medical and Dental College, Ahmedabad. The topic of my presentation is germ cell formation and fertilization. The human somatic cell contains 46 chromosomes, 46 being the diploid number for the cell. Two of these are sex chromosomes, the remaining are autosomes. Each chromosome is paired so that every cell has 22 homologous sets of paired autosomes with one sex chromosome from father and one from mother. Fertilization. It is a fusion of male and female germ cells to form a zygote, which commences the formation of an individual. Germ cells are required to have half as many chromosomes so that on fertilization, the original complement of 46 chromosomes will be re-established in new somatic cells. There are two types of division, mitosis and meiosis. Mitosis. It describes the division of somatic cells, a type of cell division that results in two daughter cells, each having the same number and kind of chromosome as the parent nucleus. Before mitotic cell division begins, DNA is first replicated during the synthetic phase of cell cycle so that the amount of DNA is doubled to a value as tetraploid. During mitosis, the chromosome containing the tetraploid amount of DNA are split and distributed equally between the two daughter resulting cells. Thus, both daughter cells have a diploid DNA quantity and chromosome number which duplicates the parent cell exactly. In this diagram, you can see DNA replication occurs and as a result of mitosis, two diploid cells are formed. Major events in mitosis. Cell division begins, DNA replication occurs, during synthetic phase amount of DNA double and during the distribution of DNA between resulting cells. There are four phases of mitosis: interphase, prophase, metaphase, and anaphase. In the interphase, DNA is copied. In the prophase, chromosomes pair up. In the metaphase, chromosomes line up at equator and mitotic spindle appear. During anaphase, sister chromatid pulled apart, and during telophase and cytokine, cell pinches in the middle, and as a result, two identical daughter cells are formed that are diploid in number. Meiosis. It is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells are sex cells, sperms in male and eggs in female. These four cells only have half the number of chromosomes of the parent cell, they are haploid. Meiosis involves two sets of cell division. In first division, DNA is replicated to tetraploid pages as in mitosis. In the first division, the number of chromosomes is half and each daughter cell contains diploid amount of DNA. While in the second division, it involves the splitting and separation of the chromosome resulting in four cells. In the diagram, you can see there are two phases of meiosis, meiosis 1 and meiosis 2. During prophase of meiosis 1, chromosomes pair up. During metaphase, chromosomes line up at equator, and during anaphase, chromosomes are pulled apart. During telophase 1, cell pinches in the middle, and during prophase 2, two daughter cells are formed and cells are separated from each other. During metaphase 2, chromosomes line up at equator in both the cells, and in anaphase 2, sister chromatids are pulled apart, and during telophase 2, cell pinches in the middle, and as a result, four daughter cells are formed. Meiosis involves two sets of cell division. In this diagram, you can clearly see meiosis 1 and meiosis 2. Congenital defects. Meiosis is the process occasionally malfunctions by producing zygote with abnormal number of chromosomes and individuals with congenital defects that sometimes affect mouth and teeth. Congenital malformation of the oral cavity include cleft lip palate and alveolus defect, developmental anomalies of the tongue, vascular anomalies, developmental disturbance of teeth or tooth structure, and congenital dysontogenic tumor or tumor-like conditions. Monosomy. Missing one chromosome from a pair. For example, if a female has one X chromosome, X monosomy rather than two, she has a Turner syndrome. In this diagram, you can see different appearance or facial features in patients with monosomy. Examples of trisomy. Down syndrome. People with Down syndrome may have small teeth, which can cause spacing between the teeth. They also tend to have a small upper jaw. This may cause crowding of teeth and may result in permanent teeth being impacted because there is no room in the mouth for them to come in. In the second diagram, you can see crowding of teeth. Approximately 10% of all human malformations are caused by an alteration in single gene. First, if the malformation results from autosomal dominant inheritance, the affected generally is inherited from only one parent. The trait usually appears in every generation and can be transmitted by affected parent to half of children. Examples of autosomal dominant conditions. Achondroplasia. The term chondrodysplasia includes a number of conditions that are caused by changes in the genes. They are often linked to dwarfism, which stunt the growth of many parts of body, especially the bones. Cleidocranial dystosis, also called cleidocranial dysplasia, is a birth defect that mostly affects bones and teeth. The collarbones are typically either poorly developed or absent, which allows the shoulder to be brought close together. The front of skull often doesn't close until later, and those affected are often shorter than average. Other symptoms may include a prominent forehead, wide set eyes, abnormal teeth, and a flat nose. Osteogenesis imperfecta, also known as a brittle bone disease, is a group of genetic disorders that mainly affect the bones. It results in bones that break easily. The severity may be mild to severe. In this diagram, you can clearly see a healthy bone and a brittle bone. Dentinogenesis imperfecta is a genetic disorder of tooth development. This condition is a type of dentine dysplasia that causes teeth to be discolored, most often a blue, gray, or yellow-brown color. Thank you.